Good morning to all. Yes, good morning. After heavy dosage, I think metabolic pathway is always clumsy and complex. Yeah, today I think the Jayanti Sarya, chemistry, just went for some tours outside. So some of the lectures and load is shifted to others. So today I will take your practical. Tomorrow it will be taken by the chemistry on. And here for today, we may go for a pour plate techniques for a practical session, just I'm telling. So what will be there into it? Do anybody remember? Yeah. Media, yes. Solid or liquid? Is it solid? Liquid. Okay. Pouring. So it will be liquid. Yes, pouring. Pour plate method I'm asking. So it will be a solid or the liquid? Huh? Solid. It will be liquid. Okay. Okay, fine. So while preparing, it will be a plate you are pouring or in a liquid you are going to do or in a tubes. Tubes. Petri plates. Dilution. Okay. What are the basic steps in pour plate method? So it is really difficult after 2 weeks. Okay, but we are going to prepare nutrient broth or nutrient agar. Now the question is solid and liquid, just the one important part is agar. So we are going to use the agar in pour plate method or not? Achha. Okay. So I think you are new. I'm seeing you first time. So came today first time or new joining? New joining in a class directly in second year. Jumped. How it is? From another university? Okay, from where you are coming? Okay. So if you find any difficulties, you can ask to your colleagues, otherwise come to me. So many that earlier lectures were already uploaded in our portal. You can just check it out. If this you're finding any difficulty, you can come to me. Yet they haven't started in writing of the journal. Okay, few practical sessions are yet remain. Afterward, I will ask you people to remain in a lab to write the journals before to me. It is not a otherwise it's we have the optimum time, September to October. Okay, so we can write it down our journals itself in a time. I will provide the soft copy, hard copy, everything to you. Okay, few things is required that just you need to write before to me in a journal. Everything you can write, I will provide later after a few practical sessions will be there, and it's not going to take so many days. Okay, one can write if it is the that in a one day and night maximum. Okay, but I will not looking for it just to copy and paste, just for the some sake of a writing. It will be there, but we can understood some concept while writing also even, so it will be easier for you. Why I'm doing such practice since long? That practical sessions generally maybe after or before your theory exams could be possible. Okay, now the corona conditions was there, but not now prevailing. So physical practical sessions in examinations could be possible, right? So there earlier it was very advantageous that just the Viva was given. Now the Viva will be given. Okay, but along with the practical sessions to be get done. So if required, on that time we may perform the some experiment again if required. But I think for the media preparations to till now, once again we are going to repeat few again. It will be addition in the practical sessions always, but some more or less you are going to see that it is the repetitions of the previous one. So in exams, it can be asked just to prepare the solid media or a liquid media or do inoculations, do the observations. So many points could be possible into it. About the two experiments they are going to ask, general practice. What practice is here, I'm not so sure, but two practical may be possible. So more or less it is going to remain same. So if you have written during that time, it will be far better. So in yeah, 4-plate method, what we are going to do, you people may divide in a certain groups as our usual practice is there. Okay, here it is maybe not required to have the individual plates. The simple reason is what? Here we are going to do some basic steps are into it. You require to prepare the Petri plates while pouring. So what we are going to do, major 2 or 3 steps will be there. That nutrient agar you have prepared, you have kept in the autoclave. From the autoclave, before the pouring onto the plates, we are going to inoculate it. The flask we are going to get inoculated. The inoculations, here the timing is very punctual. That when you are taking from the autoclave, it will be agar will be somewhat hot in the nature. So you have to touch with your arms, whether it is tolerable for you or not. When it is somewhat tolerable, you are going to inoculate with a given a tube. Our tips will be there. Okay, which you are going to use the tips. Earlier also you used certain tips, right? The tips and pipette will be there with us. So you may take the about a 0.1 ml as per our flask, what the density you have prepared or the volume you have prepared. Here it is might be like that. You may prepare about a 50 ml in one flask. From one flask, you are going to prepare 2 plates. So in a group, you may prepare the only 2 flasks. Okay, 2 flasks, each group is going to prepare. So 1 or 2 is going to prepare in that way. So here the one practice is what? While you are going to rotate the flask and you have to pour into given your plates. Sometimes what happen in this process or instead, yes, you are going to, I think we don't have the autoclave plates along with us, right? So you are going to wrap the plates, one step. You are going to prepare the medium for it. You are going to keep the tips also. So I will suggest in the next time also, I have asked for that tip box to be given to us so we can store it. Or else, what I will suggest in a smaller beaker or in a Petri plates, you may keep such tips additional. So sometimes in next experiment, by chance, this happen that you forgot to keep the tips. So tips will be available in our drawers. You can do another things. Sometimes we have the smaller tips are there. Individually, you can wrap in a paper. You can individually wrap into the papers and write it down on upper side. What is that? So you can store 1 ml, 5 plates you can keep. 200 microliter tips are there. So individually, if the 5 groups are there, they can also perform. Otherwise, you can pass one to another. So you are going to autoclave several things together. Why I'm telling? The reason is what? As we are coming at a 10:40, somewhere in time, we'll directly move to it. Earlier, last time, autoclave, water level, some tragedies always are there. Okay. So we have to rectify again and again. So our practical pouring is done, your part job is done. So this time will be easier. Already I have told that if you are 21 or 28, accordingly, 7 or 6 students a group can be possible. Okay. 6 students, maximum 4 to 6 you may go. So each individual group, they are going to prepare the 2 flasks. Okay. In each flask, about 40 to 50 ml. Maybe it's a 40 ml, sometimes 20 ml is there. You are finding that sometimes while pouring, if you are not having the continuous practices, then the amount is whether it's a 20 ml or 25 ml, it's not maybe maybe going to get judged. Or another problems may arise while pouring. Sometimes it got solidified within itself, right? So you are going to prepare 50 ml, group wise, 2 flasks. So every group will require 4 plates at least. So you may divide yourself at least in a 6, 6 or 7. It is up to you how you can divide. Then you are going to do a certain calculations, and the basic requirements you are going to prepare by yourself. That is the tips. Tip bags are there, right? Already in cupboard, so you are knowing about it. Whereas the pipettes are there, that also you are knowing. 2 to 3, 4 pipettes are there. I have already ordered more. But why I'm not asking more pipettes and others? Sometimes it's happened that if the luxurious things is given a lot and lots, then there is a no much importance. Okay. There is a so many pipettes are available into another labs. We can bring it. But I'm not asking them to given to us for a certain experimental part basic. That then wherever it's there, pipette is there here and there, it will be a misuse or not given a due importance. So what is the importance of each step, you can recall and remember. So that is why I'm somewhat going to restrict in every aspect, right? So basic requirements now you can recall. Any other things will be required in the power plate? Can anyone say? So this part you can write it down into you while writing the journals also. Sometimes I may provide it if you find that such basic thing is not there. So while in journal, whenever its journal is there, everything is written in your right side. Okay, that is a text is given. Left hand side is a blank page is there. So you may start with your writing your notes, protocols according to your language. You may write. If you are finding very much difficult, then I may dictate you, if required, that how to write it down in that part. Then what are the observations will there? Maybe on the same or the next page. Okay, when the result part is coming, it is advisable to have the observations before it. Maybe it's the first or second page going on. So you may draw your observations table. Last time I have shown how to draw that observations table. More or less, it is going to remain same in everyone, every places. And the writing the result will be going to remain same by performing this experiment. Okay, first point is always that by performing this experiment, you can write the name of the experiment. Stick plate method, pour plate method, estimations, whatever could be there. The amount is observed or the plate is observed having so many bacteria or the lawn growth is there. Just to write it down, that's it. No need to write long write up. Okay, if you wish to write something, you can put the another column as an observation. What you have observed. If you want to write it down so many essays, you can write. Or else on the right hand side also, here below to it, below the table, you can write it down everything, whatever you wish. So these are some basic steps we are going to follow into this. So anything else is required for doing such experiment? Can anyone recall what you will require to be given by me, or you have to ask to me? Huh? Burner will be there. Alcohol is already 1 bottle has been kept, 2.5 liter. Now you are also, everyone is knowing that it is there. Otherwise, they were roaming outside, then come to notice in cupboard. So you are going to directly inoculate in the flask itself. Yeah, if it is 20 ml is there, we do recall it's 0.1 ml. Okay, 100 microliter. So it is about 50 ml. So you may go for the 200 microliter. And we are going to observe day by day. It is good that Wednesday we are performing. So on a Thursday itself, you can see on a Friday, it's left. And Friday or Saturday, whenever you are coming, maximum on a Friday, it will require to just wash the plates. So at least we are going to keep for 24 to 48 hours. So it will be more easier for us to just observe. Sometimes happen that on a second day, some colonies maybe get entrapped. That is the main limitations. And you cannot visually check where our colonies are there. So it will require more a day to observe them. So maybe we are going to observe. In my class, I will ask some students to go in a lab to bring that plates. Okay, same again here. Friday, I am not having any lectures, but at 12 o'clock, we may go in a lab just to observe it. It became easier for us. So as the autoclaving is going on, you people manage your lunch, not a lobby. Okay. You manage your lunch, come for the pouring. As the part will be done, if time is permitted to us, I will ask to do some work. If it is not there, you can leave, but not in a lobby. Okay. It is like that. People are roaming in the lobby. It will be not going to see good. Anybody, anything is required to given to you? Just recall the pour plates method. Culture is required. So you are not asking. Okay, it will be granted that it will be given. No. Yes. Why I'm asking? In exam, I'm not going to provide the things what is been not written in your requirements. Be careful. That it is granted that you are going to take or it will be given. I'm going to check. Everything will be on a table. I'm going to check whether it is written in the requirements. You haven't written the culture. You have to perform your experiment. You have to perform your experiment. So how you will do that, I don't know. Okay. You are going to ask to some colleagues. While asking during the exam, it is generally not permitted. Okay. So it is generally not permitted. But what you can do that, I don't know. So that is why I'm telling that whenever you are going in exams, some people and something is taken as granted, it will be done or given. Okay. But here the usual practice, basic requirement if it is not written. Okay. Here it is it's called the basic requirements. Molten agar. It will be called as a molten agar. In pour plate method exam, it is asked, you have to written as a requirement as a molten agar. You are not going to prepare that medium, but you require the molten agar. So it will be our basic. That is why I'm telling that I'm going to dictate or going to ask you people to write the journal before to me. So what's going to happen during the practical time, you will realize. Otherwise, you are going to say that, sir, nobody has told such things to us. Okay, because on that day, maybe I'm examiner or may not. But the common instructions will be given to them what one has to do. Okay, so if you are going to say that, sir, we don't know about it, so no pure culture will be given to you. You have to perform your experiment. I don't know how then the colonies will grow. Okay, how you are going to inoculate it, that is up to you. But this is some basic comment. So let's move for our growth curve. Since long we are talking about it, growth of the microbes. Generally, it follows the binary fission. Growth can be only possible in microbes by binary fission only, whether it has gone through sexual reproduction, asexual reproduction, or any other mode of reproduction also. What could be the other things? In all the places, just DNA material is transferred. But the doubling, population doubling is only takes place by binary fission. So only growth is possible by binary fission. So here you can see the cells has increased in 2 to 4 to many stages are going to get formed. So if you are looking according to time, in hours we may go, that is 1, 5, 10, whatever it could be, depend upon the bacterial species, depend upon the availability of nutrients, depend upon the physical factors, it is going to move into leg to different phases. So first it is a acclimatization. Always it is acclimatization, right? If some students, yeah, she has joined our class, right? She is just going to observe some other colleagues and friends to whom I should interact. By chance, someone is going to say something, okay, I'm not going to talk with him or her next day. Okay, she is like to acclimatize in a class. Then she start to explore the things accordingly. Okay, by chance, she may find a good time. Within 1 or 2 days, she may go in a leg to lock phase, like that bacteria is also doing. Medium is given, it is inoculated, we have incubated. Sometimes happen that flask is remain idle on a table. We are waiting for all the groups to be get completed. So it is our limitations. Always I'm telling that who are running slow are limiting factor for us. For entire group, 5 groups are there. Those group is running very behind. At the last, it's become our limiting factors to do the next step, going in the next step. We cannot move without it. Yeah. If it is very late, we can do that, ask them to incubate by themselves. So leg to log phase, bacteria waiting that if 37 degrees there, I like to grow very fast. So they are waiting. So they remain in leg phase. As this get incubated, moving in log phase. See here, what is this? Time and the cell density. Number of the cells is increasing. This graph is showing the number of the cell. Binary division is going on. Then they are moving in stationary phase. In the leg phase, they are getting mature, acclimatized. In log phase, continuous division, cell division. They are moving in a binary division only. When they are moving in a stationary phase, they will attain, you can say, maturity. Now, directly they are going to cell matured first, then they are going to get metabolically active, and the other ingredient, if it is there, they are waiting for the other cells availability. Other nutrient availability. If it is get depleted, they will not going to go for the cell division. So they may go for the cell death. And the stationary phase, it is always cell death and cell divisions more or less going to remain equal. So number of the cell, here you can see the time is going on. Here it is a cell population. If it is a 1000, maybe it's a 1 lakh, 10 lakhs. At the last stationary phase, we may find that 10 lakh cells are there. It's going to remain about 10 lakhs. There may be a cell division is there. 2 lakhs going to die, 2 lakhs is going to generate it. So they will sustain. As the entry is there, exit, both the entry and exit will be balanced in a stationary phase. Some certain reason, one is a nutrients depletion. Another reason is what? Excretory or the extra waste material production. As the cell may die, they are going to release many things outside. In any system, if you have learned the thermodynamics, there is called the entropy. Entropy means a disturbance. The level of a disturbance, after a certain period, it will be there. So earlier it was a nutritive products and cell welfare. Now you may find that populations, generally if you are thinking related to populations, China or other countries in the several places, we are finding the population is rising a lot. Right. So one has to control, and otherwise they have to manage into limited space or given availability of the product. If you are going in a canteen, in a rush hour, so you will find that it is really difficult to get the one sandwich. If you are going into usual time, you can find anything. You can opt for the so many different sandwiches are there. If you like one specific sandwich, you have to wait because he or she may require more time to prepare. Just as the grill is there, it can be given directly with sauce. Or you can take the sauce by yourself. No need to ask him or her to give it. So it became easy. But the population, that the rush hour is there, not your class. So many others are also there. So you need to wait for a given time period. So it became a stationary phase, leg to log. The stationary phase, over that time, population has gathered a lot. So in here, one is the nutrition depletions, waste product formation is there. Sometimes also toxic compound is also synthesized by themselves, depend upon the bacteria. They are synthesizing some extra metabolites, which may be going to inhibit themselves. Stationary, I'm telling about stationary phase. Yeah. Everything is just I'm discussing about the stationary phase right now. During the stationary phase, what's going to happen sometimes during the leg to log phase? Cell is dividing. Okay, cell is dividing. They are also metabolically active. So they are going to release certain things outside. But it's going to get accumulated higher level at when you are moving at a stationary phase with given time period. It is suppose some another thing is there. So many times we are finding, the newspaper is also coming, that some persons are sick because of certain reason. They have kept themselves close into one given boundaries, right? So they may be going to get some food material from the outer side, but they are not disposing their waste out in back to the any room outside. So once you are getting it, every day food parcel is coming. Food parcel, if you are not going to the waste product is there, that is a plastic material, anything, you are just going to dump into one area. 1 month or after 2 months, you will find that half of the room is filled with the waste material. So surrounding the persons who is roaming over there, he is finding it difficult to sit even, because the one is like a 10 square by 10 by 10, 100 square foot, one room is there. He is getting nourished, even it is like a prisoner is there. So they have attached with the latrine bathroom in a one room. They are getting the food for the waste, they are having this. But at latrine bathroom, they can go for the washing and others. But they are not supposed to allow to throw their waste outside. What's going to happen? It is the bacteria became our prisoner by chance, because we have inoculated in given flask. They cannot throw. We have given the products. In another format, you can consider like that. One packed material is given to you in a room. Okay. When it is packed, it is fully having the space, but we are going to open it. Sauce, ketchup, or the so many things is given. One is the microwave oven is given. You can make it hot, but the waste product is not there. If you require more food, you can add. So one day it will come after 1 or 2 months. The person will feel very much problematic. Then it will going in a stationary phase. Okay. Once it is achieved that time, afterward, the person or the microbes are looking to go for a more death other than the cell division. They are going to die. They get matured once, but they are going to die after certain periods. So they are moving into the death phase. Last one is a death phase. That available your products. If it is the one room is there, by chance happen something, and you do have the certain products. I think we may heard about that. It's not sure, somewhere in a near to the China or some other countries. Before about 8 to 10 months back, that some students were gone for the sports, and the heavy rain has came, and they have moved in. I think it was in Indonesia or somewhere. That the group of students, along with his coach, they have moved into some caves in a below to the some mountain, and they have moved long and long and long continuously, and the water was reaching over there within inside in that caves. Maybe you are knowing, or maybe do you know about it? Such incidents has taken about a year back. So it was a corona conditions once, and the heavy rain was there. So caves were getting full with water within inside, and they have moved till you can imagine about a 1 to 2 kilometers they have moved inside, and the caves were like that. It is really difficult to come out for them. So many people have, for the all the governments have tried to take out the water by water pumps. They have kept the water pumps, but the water was going inside. In that condition, they do have the certain limited food with them in initial phase, and nobody is knowing about it. Afterwards, so many people have tried to find it out them, and by chance they come to know that some guys are there within that. So it has taken about 7 to 10 days to take them outer side. So food was given in a liquid or in pipeline or something. They have started to supply. Okay, food product was given. The space for them, it was about 10 by 10 feet, and 8 students were there, about 8 to 10. The chaos, the space they were sitting, it is just one smaller room. So how they have managed themselves, it is they are knowing only. Every sort of things. Really difficult to imagine. It is a very traumatic condition. So it is like they are in a like to now in stationary phase. They are going to sustain with their products. If they are having that one packet of biscuits, everyone is going to wait that till one is not completed. They are not going to open the second till they are hungry a lot, because they are not knowing on the next after 6 months what they are going to do. So in stationary phase, afterward, what happened? If nothing is going to happen, then they are going to get sick with him. in conditions. Nutrients availability within them in the solution is also not there. Storage within the cell or within the body is depleted and the cell is going into death phase. Okay, the cell is going in a death phase. So in decline, they are termed as a decline phase or death phase. Now there is no much nutrients available for the growth and they are not able to survive. So these are the basic 4 steps and it's considered as a growth curve. Curve means it is going in some curvature mode. Growth curve is considered. Okay, the same can be shown with another format. Number of the cells are given. So log exponential. Yeah, here the term has been kept as exponential. What is meant by exponential? Yeah, continuous increase. They are not going to wait for anyone. They are just dividing because they know the environment is really good and they will easily going to survive or move in this way. Stationary to prevent the death, decline phase will come. Here you can see, here wherever we are writing, we are not going to put till the 0 in the death phase. Simple, the reason could be, in our case is what? There's some cells for a given time period. Yeah, if the time is given a lot, then they may die. But within the given time period, some cells are able to survive. They will wait. They are sometimes go in starvations, but they are able to survive. It is like that in a class. Okay, if everyone is given initially one bread with butter, okay, you may survive for 2 or 3 days. But it is been you are closed for 1 week within that time, and you are going to daily give on the bread and butter for 3 days. Afterwards, nothing is given. So you will feel lethargic. On a last 7th day, if I'm coming, some just sitting or idle over the place, sleeping, because no activity is available for them because the nutrients is get depleted. But some may be more or less active. If the time is continue for 1 month a lot, what's going to happen? If you have been caught in a such way or closed in one system with your class, 30 students are there, what's going to happen? Maybe some may going to get sick or some may even die because of nutrients non-availability. One day it may come after a 25 or 30 or 70, they may die even because no water, nothing is there. At least what are the major factors? Is it just a macro? Nah, here it's a micronutrients also playing an important role. At least you are going. Sometimes you are knowing that going in a wild places, right? Maybe you are knowing Mr. Bear, always roaming, man versus wild in the Discovery Channel. If you are looking, yeah, Mr. Bear Grylls. Yeah, Mr. Bear Grylls is very well known for it. So without food, he can survive. It can eat anything he can eat and manage, but everyone cannot. But after certain time periods, one person may sick, but some able to manage by chance. So not all is going to decline. So we are not putting as a 0. Okay, we are not going to put everything at the given 0. Yeah, but after certain time periods, if you are going to continue, everyone is going to die. If the room is closed till 1 year, no one is going to survive. After one or another day, one may die. It may happen that vegetarians or non-vegetarians, one may eat to someone else, one they die even. If the certain situation is there, no possibility of the survival. Okay, one may start to eat to another also. Okay, last man may survive. On that, so many movies are coming, if you are knowing, right? So many movies are there. 36 8 hours, 120 hours. Some movies are there. Within that, the person has been closed either on certain island or on a, you can say the submarine or the boat is there. So they may start to attack one to another. For the survival, if they are knowing that how to go for 1 year, bread and butter was given for the 3 days, you have to run for the 365 days. So it is difficult. So over that time period, it may happen, everything is going in a death phase. Some may manage to maintain, they may otherwise not. Fine. So this is the some basic concepts. Everything is written. I have already uploaded. So in our case, in our bacterial community, when you are going to perform the experimental part, we will going to take the certain amount. Here the test tube is drawn. Okay. So what we may do, we may take on the initial N0 or the 0 hours is there, we will take the nutrient broth. We are going to perform into one flask. 100 ml or the 200 ml, we may prepare the nutrient broth. We are going to inoculate with 0.1 ml given culture. You are going to keep them in an incubator. Every hour, we will take the certain amount. Okay, we are going to take the certain amount. When you are going to perform this growth curve, we will prepare our nutrient broth before a day. Okay, and on a next day, as we are going to generally coming at 7:30, I will advise or I will ask group wise, at least one students may go and inoculate it. So every hour, I will going to discuss if whenever it is the practical session is there. If someone is there, you may take the permission of the respective faculty that we need to go in a lab, and you will going to do just basic step is what you need to take about a 1 ml, 1 ml, a culture or the inoculated your broth into simple one test tube, and that you are going to store in a freeze. The simple thing is what you are going to store in a freeze. Maybe for the 8 to 10 hours we are going to repeat. So at 7:30 you may inoculate it. So next reading is going to first reading is 0 hour, first at the 8:30. You can take maybe up to 2:30, up to max. I hope, right? At the 2:30 we are going to go for the measurement in spectrophotometer. Okay, so you will find here as you are moving ahead, the number of cells, the density will change, and density will be measured in optical density by using the spectrophotometer, and it will be converted. We are going to do certain calculations for it, so you can directly to indirectly you can calculate the number of the cells within the given our sample or what you have taken out, and based on that you need to plot this graph. Graphical representations will be done afterward. Okay, so I will explain on that day how to do. Just I'm giving the basic points what we are going to do, but on that day I will again repeat it because I understand after 2 weeks we generally forget what need to do, what not need to do. Okay. So there is the 4 phases are there. The same thing is written over here. I'm not going to read. Yes, this one is more important. 9:15. A few days back, I have asked about the one question. So there is NT, N0 to N. Achha, switch. Sorry. Here, N is the generation. If you are looking that, I want to go for E. coli. We know that 20 minutes is a time period for the doubling. So one generation is going to complete in a 20 minutes. Okay. So if you are working, when it is inoculated at 7:30, okay, it was been inoculated at 7:30, and last you are going to take it at 2:30. How many hours will be there? You have spent you have inoculated your flask at 7:30 a.m. You are going to take the last reading at 2:30 p.m. How many hours? 7, 5, yes, 7. 5 hours. Okay, 7, 7. 7. No idea what's going on, right? Huh? You have inoculated 7:30 a.m. You are taking last at 2:30 p.m. What is the time period between them? 7. 7. Okay, so it will be a 7 hours. Now you are knowing that 20 minutes is their doubling time. Okay, so how many doubling has gone? Generation has gone. Can you calculate? Anyone? First generation, just to enumerate more. First generation going to complete at 20 minutes. Second minute at 40 minutes. Third at 60 minutes. So each hour, 1 hour is equal to how many generation? 3 generation is going. So for the 7 hour, how many? 21 generation. Okay. So if you are looking that at the 7th or the 21 generation, what will the cell number or cell density population, probable number that you can identify using this formula? What you need to do? Initially, suppose if you inoculated 1000 cells, just I'm taking certain example that you have inoculated with 1000 and you have to multiply it by 2 raised to power. Here the how many generation is gone? 21. What will the answer that you are going to write it down? Just I'm giving the example. So earlier what I had told that how many generations is gone. So if you are looking in the step by step, suppose not like we are going to jump at 7 hours. Let's take the example of 3 hours. Okay, so how many generations will be there? 3 into 3, 9 generations may be gone. So here you can if calculate 2 raised to n0. It is a 1000 multiplied by 2 raised to 9. Now can you calculate? What will the answer for 2 raised to 9? Huh? Huh? 2 raised to power 9. 5 and 2. Or is it something else? You may need to calculate. I don't know. You have to go for how you can write it down. You can write in different formats. 2, 3, 4, 5, 6, 7, 8, 9. Yeah. So you can calculate otherwise and like this. 4 2s are now 8. Okay, is like you may going to calculate in different format. It is up to you. So what will be the real answer at this? Let everyone calculate. I don't know. Hmm? 81,000. Very good. How you have calculated? There is a history. No. It is. I'm not sure about it. How it is calculating. It will be something different. I don't know about this. What you have calculated, but. Gano, bhai, Gano. Yeah. See. Yes. What will be the answer? 2 into power 21. I have just I'm asking about this 9. Okay. Hmm? Yeah, now it's correct. I think you have made something mistake while writing. Right? 512. Okay, very nice. So everyone, I hope is getting the answer 512. So after 3 hours, 512 triple zero. So how much it is? 5 lakhs. You have just inoculated 1000 organisms. 0.1 ml is near about 1000 organism is always there. More than that, just more than 1000 organism is going to be there within it. So when you have inoculated within 3 hours, 5 lakhs. Okay. When you are coming on the next day, you are become billionaire, right? With number of the cells. Is it like a cell? If it is selling one cell on a 1 rupees, you can become a billionaire. So it is a doubling. Population is increasing a lot. So we can calculate. So when it is in exam, it has been asked sometimes. Growth curve, maybe it is been asked in the 8 marks, or just describing certain lag phase, log phase, or stationary, or 1 or 2 is there. You have to draw everything. Okay, there is a chart preparation is there that you are going to draw properly. Labeling is required, and you need to enumerate these calculations with given example. Okay, at the given time t period, how many number of cells will be there in a population? Okay, can you calculate this? Okay, so this one is the basic process into it. So I will not going to continue further more today. Just till now it is more than enough. Huh? Yeah. क्या आया 41 lakhs પાછા 10 તો 1000 તો ઉડાડવાના જ છે એમ ને પાછળના તો એમાં ખબર જ નહીં કા પછી 10 raised to 5 you can write lakhs is equal to 10 raised to 5 so 41 into 10 raised to power 8 yeah it's probably this one answer is proper number of the cells generally is coming in the 10 raised to power 8 so this is the luxury of our system to get the crores of and billions of the cells just in a given a day period. So how many things many a times when you split something on a platform knowingly and unknowingly when we are touching, right? Yesterday you have kept only a few organisms, next day you are coming with the millions. So millions of the organisms are lying and you are going to play with them. That is why I'm telling that we have to take care about it. If it is many hospitals or the so many places are pathogenic areas are there, we never know. Very much hygienic and non-hygienic things. It has been observed. Yeah, currently if you are going in the computer labs any time or general places are there, those who are working, see in others highly contaminated place, it is their mouse and the keyboard in a lab because everyone is coming. Whenever it's slimy, sneezing, don't know, yes, sorry, yeah, directly coming from the washroom. Many a time is not there. Directly coming from the canteen. Tissue paper is given or not been used, cloths, something we are going to do. This is usual, and we are always going to touch. Keyboard, one has to do. Even the switchboards, even our door lock. Anybody has seen anyone to clean the switches any time in lifetime at your home even? No. Why we are not doing? Never know about it. Right. So these are the basic issues and problem into it. So further, I'm not looking to move ahead. So you can put your attendance in the given box. We are going to meet around at 10:40, 10:45 in 515. Okay, we'll do afterwards. You may divide yourself. And further things as this morning time I have told, we will perform. As you are going to inoculate, pour is done, inoculation is done, you people will be free. So how you are going to handle autoclave and others, it is up to you people. Right. Yes. Generation. Yeah. It is the first generations require 20 minutes. Second generations will come at a 40 minutes for the equal line. To get double, yeah, I don't know. Someone has told that 2 raised to power 21 is 41 lakhs. I don't know. CR has told. You can ask to him.